Hello everyone. Welcome to BioTecNika's Vijeta Series 2022, which is specifically targeted for CUET UG. Right. So this series is going on from 28th June. It will go up to 13th July, and the timing is 6 to 7 p.m. Now, on the previous class, that is which was held on first, that is last Friday, we covered the topic of Mendel's principles, right? That fell under Unit 2, Genetics and Evolution. And within the same unit of Genetics and Evolution, today, what we are going to see is the next immediate topic, which is deviations from Mendelism. So, Mendel's principles is the core, it's the basics of all hereditary inheritance mechanisms, but that is not applicable to inheritance of all other types of characteristics and traits. It is not universally applicable. So when scientists were studying Mendel's principles, they tried applying his principles to different types of organisms for different characteristics, and they found that the laws were not directly applicable as it was not applicable. So they devised deviations from Mendelism. You can also call it extensions of Mendelism, right? So today let's look at these deviations or extensions of Mendel's principles. The very first one is related to dominance. Now, if you remember in the previous class, what we studied was the concept of dominance, that there are two alleles. If the two alleles are different, then there is one allele which is dominant over the other one, right? So for the allele for roundness was completely dominant over the allele for wrinkled, or allele for yellow was completely dominant over the allele for green, right? That is why in the heterozygote, in a heterozygous situation, you could only see the round phenotype, or in a heterozygous situation you could only see the yellow phenotype, that is the dominant phenotype. Now, there are deviations from this particular principle. What is this? The first one is something called as incomplete dominance. What is incomplete dominance? It simply means partial dominance. That means the dominance is not complete, it is not a full dominance. Partial dominance, in which a gene is unable to express fully and it shows only a partial phenotypic effect, right? For example, normally if we consider the red phenotype, the red color of the flower, that is the phenotype that we are talking about. The trait is the color of the flower. So here, under normal circumstances, we would have assumed red to be completely dominant over white. Okay, let's assign this capital R and let's assign this small r. If red was completely dominant over white, then in the heterozygous state, you should have only seen the red phenotype. That means if the plant producing red flowers was mated with the plant producing white flowers, you should have only seen red flowers in the F1 generation, right? But in certain instances, that is not the case. This has been seen, for example, in a snapdragon plant. In snapdragon plants, the allele for red flower is dominant over the allele for white flower. Okay, let's assign them capital A and small a. Okay, as per the diagram. So capital A is the dominant allele, small a is the recessive allele. Now, what would you expect under normal circumstances? You would expect capital A, that is homozygous dominant, to be red in appearance. You expect small a to be white in appearance. And if you would have followed Mendel's principles, if the example was following Mendel's principles, then the heterozygote should have been red. That is, it should have shown the dominant phenotype. But in snapdragon, that is not the case. Homozygous dominant definitely shows red, homozygous recessive definitely shows white, but the heterozygous condition, the heterozygous genotype, that shows a phenotype which is intermediate between that of the two parents. right? So these are the two parents, the plant producing red flower, the plant producing white flower. They are the two parents. And the heterozygote, which has been the F1 generation, which is produced by mating these two, is producing pink colored flowers. So now this pink phenotype is not matching any of the parents, right? It's neither matching the red parent, it's neither matching the white parent. So this pink phenotype is somewhere intermediate, and since it is intermediate, it is not showing either the red phenotype or the white phenotype. We consider this an example of incomplete dominance. So what is incomplete dominance? It's partial dominance in which a gene is unable to express fully and shows only a partial phenotypic effect. The heterozygote, see, this is the heterozygote. This heterozygote shows an intermediate phenotype, in this case the pink color, that falls in between the dominant and the recessive traits. Red is dominant and white is recessive. So in snapdragon plant, the allele for red flower is definitely dominant over the allele for white flower. But in the heterozygous condition, the red color is not fully expressed. This results in pink colored flowers. Now, what shade of pink it is? That can be any grade, right? So, can you see this is considered complete red, this is considered complete white, right? And you can see all these intermediate colors ranging from slightly red to very close to white. So these are the range of colors that fall in between red and white. So any of these can be considered as pink. Okay, I hope you understand. If the phenotype of the heterozygote is showing any of these colors between the two parents, then it is considered as an incomplete dominance. Now, why is this happening? It's possible that it is because of the enzyme. So these alleles represent alternative forms of the genes, right? So the dominant allele and the recessive allele are coding for something, they are making something. So the dominant allele makes a normal enzyme, and if normal enzyme is synthesized, then it will give rise to red pigment. So there's a precursor, it is converted into the red pigment, and to have this conversion, to make the red pigment, you require a fully functional enzyme. Okay, so this enzyme is coded by this dominant allele, and if it is the recessive allele small a, it makes a defective enzyme. Either the enzyme is not synthesized at all or it is defective. So this defective enzyme is not allowing the synthesis of the red pigment, and that is the reason why you are getting an intermediate color in the heterozygote. Let's take another example of this same concept of incomplete dominance. This example is different. It is the fruit color of eggplant or brinjal, right? Now, in this, now we are talking about the color. Okay, so the characteristic you're studying, the trait you're studying is the color of the fruit. So you have the parental generation where one of the parent plant is producing purple fruits and the other parent plant is producing white fruits. The genotype of this parent is capital P, and this one is small p, small p. So this is homozygous dominant, this is homozygous recessive. So when a homozygous plant that produces purple fruit, capital P, is crossed with a homozygous plant that produces white fruit, small p, what will be the genotype of the F1 progeny now? This parent plant will produce gametes containing the capital P allele. This parent will produce gametes containing the small p allele. And during fertilization, when they are mating with each other, fertilization occurs. These two gametes fuse together, and that is how you get the F1 generation. The zygote is formed, and that leads to the F1 generation. So, what will be the genotype of this F1 generation? Capital P allele and small p allele come together. The genotype will be capital P, small p. That means this is heterozygote. This F1 generation are heterozygotes. Now, when the F1 generation is allowed to self-fertilize, then what will happen? When the F1 generation are crossed with each other, then what will happen? Normally, what would you expect in the genotype? In genotype, you would expect capital P, capital P, capital P, small p, capital P, and small p, right? So the genotypic ratio is 1. These two come together because they are the same. So 2 over here and 1 over here. The genotypic ratio we expect is 1 is to 2 is to 1. Now, if this would have been complete dominance, what would have been the phenotypic ratio that you would have expected? If there was complete dominance over here, you would have expected the F1 generation to produce purple fruits, right? You would have expected F1 generation to show purple fruits, and in the F2 generation, you would have got purple to white in a 3:1 ratio, the phenotypic ratio. But that would have been the case if there was complete dominance. We are talking about incomplete dominance. And in incomplete dominance, the phenotype of the F1 generation is intermediate between the two parents. The F1 generation is producing violet fruits, which is not fully purple also, and of course it's not white also. So this color of the fruit is somewhere intermediate between the two parents, and that is why it's an example of incomplete dominance. Now, what will happen if you allow the F1 generation to intercross? That means if you allow these two to cross with each other, like I said, the genotypic ratio will be 1 is to 2 is to 1. But what about the phenotypic ratio? This type of genotype, okay, capital P, what phenotype it will produce? This will produce capital P, so it will produce purple, right? Small p, if this is the genotype, it will produce white brinjal. And what about the heterozygote? The heterozygote will produce violet fruits. That is the reason why, see here, you've allowed the two violet fruits to cross, right? That means the two plants producing the violet fruits to cross. So these are the gametes that are produced from both the parents. And when you allow fertilization to occur, see, a Punnett square is made over here. You will get capital P, capital P, capital P, small p, capital P, and small p, small p. Out of that, this one is definitely purple because the genotype matches with that of the P generation, the parental generation. Even small p will be white. But these two, the heterozygotes, they are producing violet fruits, right? So the genotypic ratio is definitely 1 is to 2 is to 1. But interestingly, here the phenotypic ratio is also 1 is to 2 is to 1. This is a deviation from Mendel's principle. Yes, as per Mendel's monohybrid crosses, the genotypic ratio was definitely 1 is to 2 is to 1. But in his case, the phenotypic ratio was 3 is to 1. Are we getting the same ratios over here? Well, definitely for phenotypic, no, right? Phenotypic ratio is also 1 is to 2 is to 1 in this case. That is why it's considered a deviation from Mendel's principle. So a 1 is to 2 is to 1 phenotypic ratio arises in the progeny of a cross between two parents heterozygous for a character that exhibits incomplete dominance. If it is incomplete dominance, your genotypic and phenotypic ratios will be the same. In fact, both of them will be 1 is to 2 is to 1. So when a trait displays incomplete dominance, the genotypic ratios and phenotypic ratios of the offspring are the same. The second type of deviation is called as codominance. Now, this is different from incomplete dominance. What we studied now was incomplete dominance. Now, what is codominance? In codominance, again, we are talking about a heterozygote. Okay, we are talking about a heterozygote like this, where the two alleles are different. Now, since the two alleles are different, both the alleles are going to be simultaneously expressed in the heterozygote. What do I mean by this? Let's look at the example and you will understand. In case of incomplete dominance, the snapdragon color, okay? In case of incomplete dominance, parent, one of the parents is red, the other parent is producing white flowers, and the heterozygote obtained in the F1 progeny is producing pink flowers, right? Intermediate color. What if we consider an example of same, the color of the flower, but now the phenomenon is codominance, it is not incomplete dominance. So now this example is in rhododendron flowers. This particular parent is producing red flower, this particular parent is producing white flowers. If you cross them, if you cross these two flowers and you look at the phenotype of the F1 progeny in codominance, you will find that both the alleles are expressing themselves. Okay, this parent is capital A, capital A, this parent is small a, small a. The F1 is heterozygous, capital A, small a. The capital A allele codes for the red color, the small a allele codes for the white color. And that's why in heterozygous condition, both red and white are expressed. The capital A is also expressed because that leads to red color. Small a is also expressed, that leads to white color. So what is codominance and how is it different from the first one, that is incomplete dominance? In codominance, both the alleles are simultaneously expressed in the heterozygote. The heterozygote simultaneously expresses the phenotype of both the homozygotes, right? These two are homozygotes, both their phenotypes are expressed in the heterozygote. So you can see parts which are red and parts which are white. There is no intermediate expression of the two homozygotes, there is no blending of the phenotype. Okay, if this was the case of incomplete dominance, your entire flower should have been pink in color. If the red parent is crossed with the white flower producing parent, your entire flower should have been pink. The entire flower should have shown an intermediate expression. That would have been considered as blending of the phenotype. But you're not seeing blending of the phenotype here. Both the alleles are expressed. Okay, so there is no intermediate expression of the two homozygotes. I hope you've understood the difference between the two, that is the incomplete dominance and codominance. So rhododendron flowers which are heterozygous, that is both the alleles are different, this will express both red and white color instead of the dominant red or intermediate pink color. The third type of deviation as per the Mendel's principles is multiple alleles. Now what do you mean by multiple alleles? As the name indicates, there are more alleles, but where exactly are these more alleles present? Definitely not in one individual. Normally when we study genetics, etc., we are talking about diploid organisms. Okay, we are all diploid organisms. Diploid organisms have two alleles because we have two sets of chromosomes, right? All diploid organisms have two sets of chromosomes. One set we have got from our mother's side, the other set we've got from our father's side. So a diploid organism will always have two alleles at any given locus. But then how are we talking about multiple alleles then? What is where does the question of multiple alleles come into the picture? So we are not talking about one organism, we are talking about a population. So you have a population of individuals, and within that population there are multiple alleles which are circulating. Okay, so there are some genes which show more than two alleles circulating in the population, and that is the concept of multiple alleles. Let's look at the example of ABO blood group. Right now, in the earlier examples, say in Mendel's examples, when he was studying the color of the seed or the texture of the seed, there were only two alleles, right? There was round and one which codes for wrinkled, or there was one which coded for yellow, one which coded for green, so on and so forth. In the population of humans, there are three alleles which are involved in the blood grouping determination. So these three alleles are IA, IB, and I. This is written as small I. Okay, so there are three alleles in the human population, but humans are diploid. That means at a time one individual can have only two of these three alleles, right? So in the population there are three alleles, but an individual will have two of these three alleles. So now that means if two of these three alleles can be present in an individual at a time, that means combinations are possible, right? Three alleles, and how many combinations are possible? We can have six combinations. How? We can have IA together. Okay, so this is homozygous for IA. We can have IB together. This is homozygous for IB, and homozygous for the small I allele. Apart from that, then there are three combinations: IA and IB together, IA and small I together, IB and small I together, right? So an individual will have two of these three alleles, and this will determine the individual's blood group. So how do the six combinations give rise to individual blood groups? IA allele codes for the antigen A, IB allele codes for the antigen B, and the small I allele does not produce any antigen on the RBCs. Where are these antigens present? On the RBCs. So if this IA is coding for A antigen, IB is coding for B antigen, and the small I does not code for any antigen. Now, how is the relation between these three alleles? The relation is this way: IA is completely dominant over small I allele. IB is completely dominant over small I allele, whereas IA and IB alleles are codominant. Now, what do you mean by this? That means if IA and small I alleles are present together, what will be expressed? What antigen will be expressed? A antigen will be expressed. If IA and IA are present together, again A antigen will be expressed. See, like this: homozygous dominant IA, this is A antigen. IB codes for the B antigen. Okay, if IA is present along with small I, it still codes for the A antigen. The reason is IA is completely dominant over small I. If IB is present with small I, it will code for the B antigen because IB is completely dominant over small I. Now, what about the remaining two? If the two small I alleles come together, then there is no antigen on the RBC at all, and that gives rise to blood group O. But what if IA and IB are present together? Then what? IA allele and IB allele are codominant, and we have studied in codominance that both the alleles will be expressed, right? That is what is codominance. Both the alleles in a heterozygote, both the alleles are expressed. So here IA is also expressed, IB is also expressed. So on the surface of the RBCs, you will have A antigen also, and you will have B antigen also, and that is why the blood group becomes AB. Right? So this is an example of codominance within the concept of multiple alleles. Multiple alleles is nothing but more than two alleles in circulation within a population. Remember, I'm stressing on that because it's important. Multiple allele concept is applicable to a population, not individuals. Right? So a person with blood group A only has A antigen on the RBC. Person with blood group B only has B antigen on the RBC surface. AB blood group has both A antigens also and B antigens also, and blood group O means there is no antigen on the surface of the RBCs. So specifically here, IA and IB are completely dominant over small I, and IA and IB are within themselves, IA and IB are co-dominant. Right, so let's just quickly look at an example of how inheritance of these alleles occur. So here is the father whose blood type is A, and in the diagram they have clearly given that this individual is heterozygous. He is not homozygous for A, he is heterozygous. That means the genotype is IAI. Okay, so that is A and O, indicating this is present IA and the other one is small I. The mother is blood type B and she is also heterozygous. That means her genotype would be IB and I. Now, what are the possibilities of the blood groups to be inherited in the children? In that case, if IAI, IB and small I. Now, if IA and IB come together, if these chromosomes come together in the zygote, then in that case you will get AB blood type. Okay, you will get a child with the blood type AB. What if IA from here and I from here come together? Then you get a child with blood type A, this one, IAI. What if I and IB come together? In that case, you will get a child with blood group B, which is heterozygous IBI. And what if the two I alleles come together? Then the two small I alleles come together, small I, small I. In that case, you get a child with blood type O. Okay, so this is the inheritance in case of the this example of blood grouping. Remember, in an individual, two alleles are present because we are all diploid, but within a population, you have multiple alleles. Now, like I said, within multiple alleles concept, specifically IA and IB are codominant, right? So just the AB blood group, not the others, okay? Not A, not B, not O. Just the AB blood group, just this blood group is an example of codominance. So individuals with blood group AB are heterozygous with two different alleles, IA and IB. In these heterozygous individuals, both the alleles are expressed. So this will produce A antigen on the surface of the RBCs. This will lead to production of B antigen on the surface of the RBCs. And that is why that blood group is called as AB. The next concept is lethal alleles. Now, what do you understand by the term lethal? Lethal indicates dangerous, right? Something which is harmful. So these alleles or these genes are exactly that. They are alleles that cause the death of the organism that carries them, right? That is why they are called lethal alleles or lethal genes. That means if these alleles or genes are present in the organism, it will lead to their death. It can be fatal for that organism, for that individual organism. Now, lethal alleles can be fatal in the homozygous condition or in the heterozygous condition. Okay, let's say small a is the lethal allele. That means if this is the lethal allele, just giving you a random example, okay, a brief example. So in homozygous condition also, in certain cases it can be lethal, or it can be lethal in heterozygous condition also. That means if only one copy of that small a allele is present, that can be considered lethal, that can be lethal. Or in homozygous condition, if both the copies are small a, then it will lead to lethality. Let's see an example and then you will understand better exactly what are lethal alleles, why do we have them, why are they still present in our genome. Well, one of the examples is inheritance of coat color in mice. Okay, now in mice, yellow is dominant over gray coat color. Okay, in actual terms it is called as agouti, but I've just written gray for easier understanding. So yellow is dominant over gray coat color. That means capital Y is dominant over small y. Now, what would you expect if you cross two heterozygous mice? Okay, this is yellow mice. This is also yellow mice. Both of them are heterozygous. See, yellow mice, both of them are heterozygous. Now, if it was simply following Mendel's laws, Mendel's principles, what would you have expected in the progeny? This parent is going to produce two types of gametes. This parent is going to produce two types of gametes, right? And after after fertilization, what would you expect? You would expect capital Y, capital Y, capital Y, y, and small y. Yes, these are the three genotypes that you would expect. If it was normally following the Mendel's laws, Mendel's principles, the ratio would be 1 is to 2 is to 1, right? And what would be the phenotype? What would be the phenotypic ratio? This definitely would have been yellow. This also would have been yellow, and this would have been gray, right? So your phenotypic ratio would have been 3 is to 1, 3 parts yellow, 1 part gray. But interestingly, the allele for capital Y is lethal. So when the two Y gametes come together, that means the two gametes containing the capital Y allele, when they come together, that leads to a dead embryo. That means there is a possibility of fertilization, there is a possibility of implantation. The embryo will grow to a certain extent, but it will be aborted. It leads to early abortion, right? Now why does this occur? This only occurs if the capital Y allele comes in the homozygous state. If it comes in the heterozygous state, for example, this is heterozygous, right? The capital Y has gone with the small y. So in the heterozygous state, it is not lethal. The mice survive. They are yellow in appearance, coat color is yellow, and they survive. And if small y and small y come together, it is the gray or the agouti color, right? So why are the homozygous dominant mice dying? Why are they not being born? It's because in the homozygous condition, the capital Y allele is lethal, right? So yellow is dominant over gray coat color. A cross of two heterozygous yellow mice initially produces a genotypic ratio of 1 is to 2 is to 1, right? 1 out of 4 are capital Y, then about 2 out of 4 are capital Y, and 1 out of 4 are small y. So there is a genotypic ratio initially, but these mice, the homozygous dominant capital Y, they die early in development, and that is why they do not appear in progeny. The embryo would be aborted in the early stages. The that particular mice will not be born, and in the progeny you will only see yellow and agouti, but now the ratio will change. How will the ratio look like? The ratio will be 2 is to 1. You should have got 3 is to 1. Okay, as per Mendel's, you should have got 3 is to 1, but because of the presence of the lethal allele, the ratio changes to 2 is to 1. 2/3 of the progeny are yellow, and 1/3 of the progeny are agouti or gray. The next concept is polygenic characteristics and pleotropy. Now what do these two terms imply? Normally when we study gene, genetics, heredity, etc., we have this basic assumption that one gene is is only responsible for one effect or one characteristic or one trait, right? We are all guilty of that. This is what we understand always, that there is one gene which controls one trait, one characteristic, or one effect. And there are genes which do this, but this is not the case all the time. It's possible that many genes interact with each other and they overall contribute to one single effect or one single trait or one single characteristic. So such characteristics are called as polygenic characteristics or polygenic trait. What is polygenic characteristics or polygenic trait? Many genes contribute to a single effect. Characteristics which are encoded or controlled by genes at many loci. For example, skin color. Our skin color is not controlled by one gene. There are multiple genes. Eye color, hair color, our body shape, height, weight. Apart from multiple genes, they are also influenced by environmental factors, right? So these are characteristics which are not controlled by one gene, but there are multiple genes at multiple loci that are controlling these characteristics. For example, eye color. Okay, if you just try and find out how many genes control eye color, well, you will find varying numbers, but the common number you will find is 15 to 16. Okay, there are actually within 15 to 16, there are two of them which are important, but overall, can you imagine, there are 15 to 16 genes at different loci which are controlling the eye color. So your final phenotype, final eye color, is actually being controlled by these many genes, right? So this is an example of polygenic characteristic. Now what about pleotropy? Pleotropy is kind of opposite. If there's one gene which is affecting multiple characteristics or multiple traits. Okay, so you're talking about one gene only, but it is affecting or it's controlling multiple characteristics, multiple traits. So it's kind of the opposite of polygenic. Okay, kind of opposite of polygenic. So this is pleotropy. This concept is called pleotropy, where a gene has multiple effects. Now, the example in this is, let's take the same example that we discussed just now, that is lethal alleles. Lethal alleles, you saw that they are responsible for the yellow coat color in mice, right? The capital Y allele. But why does it lead to the death? Why having homozygous capital Y? That means why having two capital Y allele leads to the death? It's because this particular allele definitely it controls the coat color. This allele is definitely responsible for yellow coat color. Okay, so yes, this gene capital Y is controlling one effect, which is coat color. But apart from that, this particular allele also has control over multiple effects, like causing diabetes like condition, you know, diabetes. The second one would be obesity and propensity for development of tumors. So can lead to tumor formation, right? So that one Y allele, that capital Y allele, which is primarily responsible for coat color, also is controlling other characteristics, other traits, other effects, like leading to diabetes like condition or obesity or tumors. And it's because of these that the homozygous capital Y dies. It's because of these that mice having capital Y genotype is going to die early. It's not the yellow coat color. Okay, it's not that the yellow coat color is causing the death of the mice, no, but it's because of the presence of this allele which is influencing other characteristics, which is leading to the death, right? So this is polygenic trait, where many genes are contributing to one effect, one characteristic, one trait. And pleiotropy, where one gene is influencing or it's controlling multiple effects. Now, in actuality, within our genome and within the genome of other organisms, you have both of them working together, right? So for example, this gene is undergoing polygeny also, right? This gene is undergoing polygeny also. That means there are multiple genes which are controlling the same effect. The 1, 2, 3, 4, all the genes are controlling the same effect. So it's undergoing polygeny also. But at the same time, it is also undergoing pleiotropy. For example, this one gene is controlling multiple effects also. So in actuality, within our genomes, within genome of other organisms, you have both of them working together. Okay, polygenic traits and pleiotropic traits can overlap with each other. Yes, so the next one is sex chromosome inheritance. Even the inheritance of sex chromosomes does not follow the tenets of the Mendel's principles. So sex chromosomes do not follow Mendelian pattern of inheritance as seen in the autosomes. What are autosomes? The other chromosomes which are not sex chromosomes. So autosomes are non-sex chromosomes. Autosomes do follow the Mendelian pattern of inheritance, but not sex chromosome. If you consider autosomes, well, autosomes are considered all the autosomes. Like, let's say we have 22 pairs of autosomes. Okay, humans have 22 pairs of autosomes. Now these 22 pairs, all of them follow the Mendelian pattern of inheritance. That means each pair will have chromosomes which are homologous pair. They make a homologous pair because they are similar. They carry the same set of genes, right? So if there is a gene here which is controlling eye color, that means on the same locus on the homologous chromosome, the gene will control eye color, right? If there's another gene here which is, say, controlling some, I don't know, the flower color. Okay, there's one gene here which is controlling flower color. Then simultaneously on the homologous chromosome also there is a gene which controls flower color. Okay, so autosomes, they have this. They have the genes on both the chromosomes of the homologous pair. They are the same, but not sex chromosomes. There are two sex chromosomes and they are different. In humans and in other majority of the organisms, actually we follow the XXXY pattern. So in us there's the X chromosome and the Y chromosome. Can you see here? This one is the X chromosome, this one is the Y chromosome. X and Y chromosomes are definitely different in size. Can you see? X chromosome is much bigger as compared to the Y chromosome, which is much smaller. In fact, their centromere position is also different, right? This is submetacentric. Your X chromosome is submetacentric. Your Y chromosome is acrocentric, right? And more important than that, the genes that are present on the two chromosomes, the genes that are present on the X chromosome are completely different from the genes present on the Y chromosome. So X and Y chromosomes are different in size. The type and the number of genes on the two chromosomes are also different. Females have two X chromosomes. See, this is female, two X chromosomes. Males have one X and one Y chromosome. This is a male, one X and one Y chromosome. So how will the gamete formation take place? During the gamete formation process, the male produces two types of gametes. That means among his gametes, 50% would contain the X chromosome, 50% would contain the Y chromosome. For the female, all 100% of the gametes contain the X chromosome. Can we make a Punnett square for this? If we make a Punnett square for this, X and X is written over here. This is from the female side. This is from the male sperm, 50% X and 50% Y. Now when fertilization occurs, here you have XX, here you have XY, this is XX and this is XY, right? That means 50% possibility of having a girl child, 50% possibility of having a male child. So the sex ratio that is produced is 1:1. One male is to one female. And what chromosome is it that determines the sex of the progeny? It is the Y chromosome. It's the presence or the absence of the Y chromosome which determines the final sex of the progeny, right? So that is why the inheritance of sex chromosomes is considered to be a deviation from Mendel's principles, because both the chromosomes are different. In all the other 22 autosomal pairs, the two chromosomes are similar. That's why they are called homologous pairs. But X and Y cannot be called homologous pair. They are not homologous. They are not same in size, shape, centromere position, or the genes that they are carrying. So difference in the combination of sex chromosome leads to different sexual morphology and characteristics in the two sexes. Because if two X chromosomes come together in the progeny, it's going to be a female. If one X and one Y comes together in the progeny, then it is going to be a male. So what combination the chromosomes are coming in the progeny, that determines the sex of the progeny. It determines the sexual morphology and characteristics of the progeny. The next one is epistasis. Now, epistasis is different from the others that we studied earlier, because epistasis involves interaction between two different gene loci. All the other examples that we studied earlier, that basically was for one gene. Okay, what happens at one gene? If at one gene, if two different alleles are present, how do the alleles interact with each other? That was more of allelic interaction. Epistasis is an example of two gene interaction. So if there are two or more genes at two different loci, then how do they interact with each other? That's one of the examples, is epistasis. Now, epistasis, what does this mean? Like I said, it is a type of an example where there is interaction between two or more different genes. Specifically, in epistasis, one gene masks or hides the effect of another gene at a different locus. Okay, so there is one gene which is masking, or it is hiding or suppressing the effect of another gene which is present at a different locus. The example for this, one of the examples, by the way, there are different types of epistasis. I'm just giving you one example of a type of epistasis, which is coat color in Labrador retrievers. Now you can see three different colored Labrador retrievers here: black, brown, and yellow, white, whatever you want to call it. Okay, I've written yellow over here. Now, the final coat color, the final coat color of that dog, Labrador retriever, is determined by two loci. There are two loci which determine the coat color. That means there are two loci, two different genes which are interacting with each other to determine the coat color. At the first locus, the two alleles that are present, capital B and small b, they determine the color of the pigment. Okay, they determine the color of the pigment. Now, at the second locus, which is locus 2, there are two alleles, capital E and small e. But what is determined over here is not the color of the pigment, but whether the pigment will be deposited or not. Okay, whether the pigment will be deposited or not. The color of the pigment is determined at locus 1 by capital B and small b, and whether the pigment should be deposited or not, that is determined by another locus, that is another gene, by capital E and small e. So capital B allele is dominant, it codes for black pigment. Small b allele is recessive, it codes for brown pigment. At the second locus, capital allele E is dominant, it allows the pigment to be deposited, whether the pigment is brown or whether the pigment is black. But whatever the color of the pigment is, but it will be allowed to be deposited if it is capital E. But if it is small e, which is recessive, in that case, there will be no deposition. And if there is no deposition of the brown or the black pigment, then the coat color will be white or yellow. Right? So, what will be the different types of genotypes then? If the genotype is capital B dash, capital E dash, capital B indicating that it is black pigment and capital E indicating that it will be allowed to deposit. So, the final phenotype will be black. What about the second combination, small b, capital E dash? If it is small b, that means it is recessive, that means it will encode the brown pigment. And it is capital E over here, so that means pigment will be allowed to be deposited, so it will be brown. What about the third one? The third one is capital B dash, small e, small e. Now, it is small e, small e. So, it doesn't matter what allele is present at the first locus, right? In the last two cases, at the second locus, it is small e, small e. So, it does not matter what is present at the first locus. It is not going to allow the deposition of the pigment. It is going to be yellow. So that is why this is an example of epistasis, because the genotype at the second locus masks the expression of the genotype at the first locus. Right? If small e is present, it does not matter what is present at the first locus, whether it is capital B, whether it is small b, it is not going to allow the deposition of the pigment. Right? The next one is linked genes. Even linked genes don't follow the inheritance pattern as given by Mendel. They don't follow Mendel's principles. What is linkage? The term linkage is used to determine inheritance of genes which are located on the same chromosome. Right? See, for example, look at this one. This is a single chromosome where gene 1 and gene 2 are present kind of close to each other. Yes. So these two genes are said to be linked. Now, here in the second case, gene 1 and gene 2 are present on two different chromosomes altogether. Okay, so they are not linked. In the third case, gene 1 and gene 2 are on the same chromosome, but they are at distance from each other. There's quite a few distance between gene 1 and gene 2. So in this case also, though they are present on the same chromosome, but you consider it not linked. Okay, so both these are not linked, whereas if the two genes are present close on the same chromosome, you consider it linked. So linked genes tend to be inherited together, and they do not obey Mendel's principle of independent assortment. Linked genes don't follow the principle of independent assortment, whereas unlinked genes do. If the genes are not linked, they will follow the principle of independent assortment. Okay, now in the previous class we did study independent assortment, and I hope you remember. Now there are two genes over here. Okay, capital A small a, capital B small b. These represent two different genes. Now during the gamete formation process, four types of gametes are produced. Why four types of gametes are produced? Why not two? That's because of the allowing the independent assortment. Okay, capital A can go with capital B, small a can go with small b, but the other combination is also possible. Remember the second possibility, small a can go with capital B and capital A can go with small b. So four types of gametes are possible, and you make a Punnett square with the four types, you get the 16 squares. Okay, this is just a representation of the dihybrid cross. I'll not go into the details of that. But what will happen if these two genes are on the same chromosome and they are so close together that they are linked? In that case, okay, if A and B genes are linked, then during meiosis, during the gamete formation process, they will not separate. They will not undergo independent assortment. So you will get only two types of gametes, which is capital A, capital B, and small a, small b. You will not get these combinations. Why? Because the two genes are linked. They are present on the same chromosome and they are close to each other. So now in this case, if you make a Punnett square like this, you only get four possibilities, which are similar to that of the parents. So you don't get new combinations over here. Right? So this is linked genes. They don't follow the principle of independent assortment. Now, second last one is cytoplasmic inheritance. You know that a zygote is created after fusion of the sperm with the egg. Now, specifically when I say fusion, it's the nucleus of the sperm which enters inside the egg or the ovum. Okay, not much cytoplasmic content of the sperm enters inside the egg or the ovum. So the cytoplasmic content of the zygote is actually from the egg or the ovum. That means the cytoplasmic content is inherited from the egg. For most organisms, all the cytoplasm is inherited from the egg. So these cytoplasmically inherited traits are always passed from the mother to the offspring, never from father to the offspring. Right? One example of that is the mitochondrial traits. You can see this is a cell and you can see these mitochondria organelles. Mitochondrial inheritance is always from the mother's side. So you can see the example over here. Let's take the example of inheritance of mitochondrial diseases. These are the diseases or disorders which arise from mutations in the mitochondrial DNA. One example is LHON, which is Leber hereditary optic neuropathy. Now, in this case, there is a mutation in the gene which codes for NADH dehydrogenase enzyme, which I'm sure you know plays a role in the electron transport chain of respiration. This leads to rapid loss of vision in both eyes, resulting from the death of the optic nerve. Now, more important than all this is how is this trait inherited? If the mother is affected, then all her children will be affected, irrespective of the gender of the child. Okay, 100% of the children will be affected if it follows the mitochondrial pattern. So if you're talking about the LHON disease, yes, if mother is affected, all her children will be affected. But what if father's mitochondrial DNA is affected? Will his sperm mitochondria be transmitted to his children? No. Children get their mitochondria from their mother's egg or ovum. So if father is affected, no problem, because that it's not going to be transmitted to the children. There is no transmission to the children. All the children, whether it is girl child or boy child, all children are unaffected, right? So, guys, with this, we come to the end of this topic. I have given some questions towards the end, but since I'm running out of time, this will be a part of the video that is in front of you, and you can go through it. The questions have been based on the topics that were covered earlier. So, you can go through these questions, you can try and answer them yourselves. Just 3 questions are there. All right. So if you have any more doubts, any information you want, you can email, you can contact toll free number, you can give your feedback or testimonials. And please do download the BioTecNika app for more information, for courses, etc., that are available, and resources. You will get a lot of resources there. All right. Thank you so much for your time. Yeah. See you next time. Bye bye.